The data sharing that we do at CPath involves aggregating high quality, preferably controlled clinical trial data that can be fully aggregated and curated across, and its data across multiple drug development programs. And the value of that is it gives us enough information to answer a specific objective. We typically are sharing data and aggregating large data sets to provide evidence for a new tool, like a biomarker or a clinical outcome assessment instrument, or to develop a disease progression model. And that's where the value comes in the sharing of data, because everyone who's developing drugs can benefit from the knowledge we gain from that disease progression model. I believe that sharing data for the purposes of what I've described, when we're trying to achieve some common research objective, does, in the end, decrease cost of clinical trials. So for example, if we have a disease progression model that quantifies the impact of the disease, the drug, and the trial design, companies can simulate different trial designs in a model-based approach rather than having to go out and actually conduct 4, 5, 6 different kinds of trials. Therefore, we have more confidence that they are putting the right patients in the trial. They're studying the right phase of the disease. All of that means you would enroll fewer patients, and that brings the cost down. And hopefully, if the trial is designed better, it might be shorter, which also is a cost saver. In tuberculosis, CPath has been working for some time in a collaborative environment with the Bill and Melinda Gates Foundation and TB Alliance. And we realized in this small TB research community, we need to learn from each other. CPath created a clinical trial repository where we are encouraging all organizations that are conducting trials to share their data in that repository so that we can learn why drugs fail or why a study is inconclusive, because there have been a number of those. We have a relational sequencing database in which we capture targeted sequences from the genome of the isolates that cause TB so that we can better understand what might be causing these isolates to become resistant to treatment. When we can, we have the phenotypic data of the patients that we also map to that isolate. The goal of all of this data sharing is to better understand how resistance develops in particular drugs, and eventually this becomes a surveillance tool for monitoring resistance and could potentially be a clinical utility of a physician being able to understand what drug or drug regimen that particular patient is most likely to benefit from.